Well, I'd like to introduce, with great pleasure, Rod Wilson, our final Bevan speaker. He obtained his BSc and his BSc Honours in Biological Sciences and his PhD in Fish Physiology and Ecotoxicology from the University of Birmingham in the UK, and has published as many as 65 scientific papers already. Amongst his many achievements, he was awarded the 29th Annual Rosenstiel Award for Outstanding Achievement and Distinction in Marine Biology, Biological, Oceanographic, and Fisheries Science. And since 2000, he's also filled his spare time with being the assistant editor of the Journal of Fish Biology. He's currently a professor in integrative animal physiology at the College of Life and Environmental Sciences at the University of Exeter. He's a wide-ranging scientist, but really at the core, he's a comparative physiologist. He does research that focuses on how animals maintain their internal chemical balance in the face of environmental variability. Particularly, how they develop mechanisms to cope with variability. His work has included research in the Amazon on freshwater fish that can even survive in pH as low as 2.5. For those that have been following what we've been talking about, that's a lot lower than we expect anything in the oceans, even in the future. Recently, and what really brought him to our attention, was his work on fish production of carbonates, and how the resulting carbonates they excrete can affect ocean carbonate balance, and also what happens with ocean sediments. And that's resulted in papers in Science, proceedings of the National Academy of Sciences, and various other top journals. Now, on a personal note, some of his friends, or perhaps I should say friends, were very kind to fill me in on a few of the bits that are just completely missing from his online resume. But it turns out that he has a real passion for Liverpool soccer, where he was born, for music, and the usual Hungarian pig offal goulash. Less well known is his love of dressing up as his favorite animal. So if you're ever at a fancy dress party and you see a giant flounder coming up to you, that's Rod Wilson. It gives me great pleasure to welcome Rod.
Thank you very much, Trevor. That could have been a lot more embarrassing than it was.
Let out those parts. Okay, we said it would be liveness.
I appreciate that. Well, first of all, this is my first time in Seattle, and it's an absolute pleasure. It certainly has been so far. I'd like to say thank you right at the start for everyone involved in organising my stay here, especially Sherry for taking me from the airport to the B&B last night. It's been a fabulous day today as well, meeting many people and such interesting conversations. It's given me no time at all to think about the talk, which might be a good thing. Anyway, it's also my absolute privilege to be part of this Bevan series, and I don't really feel worthy of finishing off the series of 10, having looked at some of the online movies of the previous speakers. I hope I can at least keep you interested and awake for the next 45 minutes in what I want to talk about, which is the rather intriguing role of fish in producing carbonate within the ocean that wasn't really known about much before a few years ago. So, this is the title that I didn't send in. It was a title that I can't remember who gave this option, decided that my title wasn't quite sexy enough, and they converted it into this, which is not bad to bring the punters in, let's face it. The title I sent in was this one. A bit more formalised. And so I wish to tell you about the surprising role of marine fish in the global inorganic carbon cycle, present and future implications, which will include ocean acidification, so don't worry, it is part of the story. And if time allows, when I finish my talk, I've got a few extra slides if questions come that way. I also might like to comment on what role it might have played in the past, so please bear that in mind for the end. So, I'm guessing that all 10 of the Bevan series speakers this term have shown a slide of the Mauna Loa atmospheric CO2 measurements. If they haven't, it'd be a surprise. Anyway, what this is all about is humans are producing more and more CO2 from burning of fossil fuels in manufacture of cement, and CO2 is rising in the atmosphere. The oceans are helping to remove some of that CO2 by absorbing it, and it dissolves in the oceans. The problem is when it does that, and goes through this reaction, it's generating extra acid or protons, which is reducing the pH of the oceans. So we're getting increased dissolved carbon dioxide in seawater, and the pH drops at the same time. Now, you might think, so what? But really, the main concern is not the magnitude, which is relatively small compared to what I've worked on, for example, in the Amazon, where fish can tolerate pH 2.5. It's not the magnitude, it's the rate at which this is happening. When you look at a geological timescale, it hasn't been happening this fast for millions of years. So the speed at which it happens is creating as big a problem, perhaps, as the magnitude for the organisms that live in the ocean and just aren't used to this kind of pH change. So it's very important to understand all the factors that control the pH of the oceans. And this is what this next slide will try to summarize in terms of the carbon cycle, or I say the carbon cycles within the ocean, because there are two. The first one is the one that most are familiar with, because this is the same as the terrestrial environment. Anything that's photosynthetic can convert inorganic CO2 into organic matter. So within the oceans, this is mainly surface algae taking up CO2 from seawater that ultimately comes from the atmosphere, converting it into organic matter. If that organic matter is not eaten and inspired, then it will eventually sink, settle out at the bottom of the ocean. And in doing so, it acts as a sink for CO2, removing ultimately atmospheric CO2, sending it to sediments in the deep ocean that over longer timescales can ultimately become sources of hydrocarbon fuels, oil and coal. The oceans are very interesting in respect to the carbon cycle compared to the terrestrial environment because they have a second half, an inorganic half to that carbon cycle. This is the process in which organisms biogenically help run this reaction, which converts calcium ions and two bicarbonate ions into a mineral called calcium carbonate, which can have various forms, but you'll notice that in doing so, it actually generates CO2 rather than taking it up. Now that's very confusing, and it confuses a lot of people that don't get the message. Obviously, there is a carbon atom in calcium carbonate, and that comes from bicarbonate ions in the seawater. But the other carbon molecule in the second bicarbonate ion ends up being liberated as another CO2 molecule. So calcification, this process of producing calcium carbonate mineral, actually contributes to ocean acidification. Now that's a scary point. That means that coral reefs are contributing to ocean acidification. That's not a message I want to get out there, really. Because I don't want it to be used by people to say, well, let's just get rid of the coral reefs. Coral reefs are important for many other reasons than that, okay? But that's important to know. Such that if these calcium carbonate particles sink to the bottom of the ocean, and they are very dense, so when the organisms that produce them die, these usually do sink, and if they reach the bottom of the ocean and become sedimented, then the net effect is a contribution to CO2 production. So understanding how it is produced and where it's going is obviously important to understanding the whole carbon cycle and this ocean acidification issue. Now, that reaction that I just showed can happen spontaneously in seawater, but it rarely does, or it happens at a very slow rate. Almost all the calcium carbonate formed in the oceans happens biogenically, induced by organisms, which might be photosynthetic or they might be animals. Here's a few examples of those, the famous ones being coral reefs, animals that make large animals that make calcium carbonate shells. On a volumetric basis, the most important are almost certainly the microscopic algae, the coprolithophores, but there are also other microscopic ones like pteropods and foraminifera. I'm sure you've heard of the rest of this term's Bevan series. It's important to know about that carbonate production from lots of points of view. One of them is a geological angle. Because any of this biogenic carbonate that's produced and sinks and sediments out can eventually, ultimately, become part of chalks and limestone rocks. This is a famous example from the UK, the White Cliffs of Dover, a very chalky rock made famous by a wartime song called "There'll Be Bluebirds Over the White Cliffs of Dover." You might know. Anyway, these can hold very important messages about climate and ocean chemistry in the past. A couple of examples of that include the fact that when you look at the coccoliths shells within chalky limestone type rocks, and you look at the amount of strontium in elements that are incorporated into the calcium of the calcium carbonate, it can indicate changes in ocean chemistry in the past on that kind of time scale. Similarly, the amount of boron included in these carbonate rocks can tell us something about the prevailing ocean chemistry, in this case pH or carbonate saturation, at the time as well. So there's a past reason to understand this as well, or to help us understand the past reason. So all of this biogenic production of calcium carbonate has been fairly well understood for a long time. And what we're now suggesting that actually fish need to be added to this list as important producers of calcium carbonate. Having said that, it's important to realise that all of these animals, or plants or algae, produce calcium carbonate as part of their skeletal material. So it's forming as a kind of mechanical function for those organisms. Our fish, on the other hand, are not. Calcium carbonate they produce is nothing to do with skeletal material. Indeed, bony fish, just like the rest of the vertebrates, including ourselves, their skeletons are made of calcium phosphate, completely different mineral, something called apatite, but it's certainly not calcium carbonate. So where's all the calcium carbonate coming from that these fish produce? Well, it comes from their guts. They're producing it inside their guts and excreting it continuously throughout their lives. It's not a skeleton that's shed every now and then, it's every day of their life. Here's a tank of schoolmaster fish from a tropical setting, put in an empty tank, you leave it for a day, and at the end of the day you'll see these little white pellets forming on the bottom of the tank. These have been excreted by the gut of the fish, settle out at the bottom because they're dense. And what they actually are is calcium carbonate crystals wrapped up in an organic mucus package inside the gut and then excreted. Here's another picture of a slightly larger flatfish, European flounder, in my lab back in Exeter in the UK. Instead of small pellets, these guys tend to produce long strings of mucus or tubes, but again, inside each of those, it's packed full of millions of crystals of calcium carbonate. If you collect those up with a pasta pipette and put them in a test tube, it looks like a bit like popcorn. Very, very white. Obviously you wouldn't want to eat this, but it's very, very white. And it's white because of all the millions of crystals of calcium carbonate within it. If you collect those, this is actually pellets collected from the first picture of the schoolmaster. This is covered in a mucus coating. If you get rid of that mucus coating, you simply treat it with bleach, you're left with aggregates of pure inorganic calcium carbonate crystals. You look at these under the electron microscope, these aggregates are fairly large. This one's about just under 0.5 millimeter long. And then if you zoom in on this, you'll see that the surface of each one of these is covered in literally millions of tiny calcium carbonate crystals. I'm going to say American football shape, they normally call it rugby ball shape. But they're all about 1 to 2 microns long. There's millions of them within each of these aggregates. Most of my physiological work, I'm a physiologist, has been focused on trying to understand why these fish bother producing all this stuff and how they produce it. That was the original questions I was asking, nothing to do with its role within the oceans. It turns out we think it's all to do with osmoregulatory physiology. In layman's terms, that's water balance. These marine fish live in a really salty solution. If you look at the blood chemistry of a teleost fish or bony fish, it's just like you and I. It's the same salt concentrations for all these different ions. Compared with seawater, that's about 3 times more concentrated. That means the osmotic pressure outside the fish is about 3 times stronger than the osmotic pressure inside the fish. That creates an enormous dehydrating force where salts will tend to diffuse in, and water is sucked out of the animal by osmosis. And in fact, I believe that this is the single most important physiological challenge that marine fish face throughout their entire lives, is the tendency towards dehydration every minute of every day of their lives. So they have to work really hard to overcome that. Now, how do they do that? Well, there's lots of things they do with their gills and their kidneys, but actually the fundamental thing is how to get water back into the blood. And of course they can't go to a bar and buy a bottle of water. The only thing they can drink is seawater. So that's exactly what they do. And they drink seawater at really quite remarkable rates. That 1 to 5 mils per kilogram per hour, the highest rate equates to an adult human drinking about 12 litres of fluid a day. So this is a lot of seawater to drink. Now, we can't drink seawater. If we do, we die very quickly. That's because our kidneys are active and they can't handle it. But seawater fish are well adapted to drinking it, and they can quite nicely absorb free water from that and remain hydrated. That's an interesting story how they do that, and I'll tell you a tiny bit about that in the next few slides. But that will be probably the deepest we go into physiology today. Now, they're drinking lots of the seawater, and the thing to point out is it contains lots of ions, but particularly I'm interested here in the calcium and the magnesium, which is relatively high levels too. What happens within the gut is at one end you've got large volumes of calcium-rich seawater going in at one end, and then you've got the excreted calcium carbonate crystals coming out the other end. How does that happen? Well, it's very simple. The cells that line the border of the intestine are continuously producing and actively secreting bicarbonate ions at really high rates. What that does is generate the right conditions to rapidly promote the reaction I showed you right at the start of calcium with these bicarbonate ions to produce precipitated crystals of calcium carbonate. As these crystals move down the intestine, they get wrapped up in this mucus kind of protective coating, and then they get excreted into the external environment. Just to give you an idea of the chemistry within this intestinal environment, it's not the stomach, by the way, it's only in the intestine. Compared here, seawater pH in bicarbonate, which is about 8.1 and about 2 millimolar, with the pH in bicarbonate in the intestine, which in marine teos is typically much more alkaline than seawater, as high as 9.2 in some species, and the bicarbonate is much higher, from 30 to 100 millimolar. These conditions are very, very supersaturated with respect to calcium carbonate, so it's bound to precipitate. And it precipitates very, very quickly indeed. One other thing to point out is there's lots of magnesium in seawater as well, and sometimes you get magnesium being precipitated with the calcium as magnesium carbonate. So a lot of these carbonates that fish are producing is quite high in magnesium. That will become important later on in the talk. So here's the deepest part of the physiology. Please don't worry about the details. So this is trying to explain why the fish want to deliberately precipitate calcium carbonate in the gut. Because let's face it, most of our tissues try very avidly to avoid that ever happening. Because when it happens in your kidneys or within your pancreas, it causes really severe damage, kidney stones, pancreatic stones, which you can actually die from. So most epithelia inside your organs are really trying hard to avoid this process happening, whereas fish are actually deliberately making it happen. So fish are dehydrated, they need to absorb as much water as possible from the seawater they drink. This is complicated. The bottom line is these are a whole host of transporters on the membranes on both sides, on the blood side and on the gut lumen cell of the intestinal cells. And the bottom line is they are there to make sure that as much sodium chloride from the seawater they drink gets absorbed into the blood. If you can do that, that itself will drive a substantial amount of water to be absorbed. Essentially, it follows by osmosis. And that's the first and primary mechanism of absorbing water from the gut. The second one we discovered a few years ago now relates directly to the bicarbonate secretion process. So there's transporters that essentially, well, there's enzymes inside the cell called carbonic anhydrase that helps promote the reaction of CO2 with water to form bicarbonate ions that are then transported into the gut lumen in exchange for chloride. I won't go into the detail of it now, but that in itself will also induce the removal of water from the gut into the blood. So that's the second important mechanism for how fish absorb water from the gut. The third one relates to this calcium carbonate reduction. Because of course, there's lots of calcium and magnesium in the seawater they drink, but little of that is actually absorbed across there. If you're not absorbed, but the water volume you're in goes down, because that water is being absorbed, then that effectively concentrates a lot of that calcium and magnesium. And the rate of water absorption is so high that you predict that these ions would get concentrated by 6 or 7-fold by the time they reach the end of the testing. Now that wouldn't be good for water absorption because that would increase the osmotic pressure here and start to pull water back the other way, which is exactly the opposite of what the fish wants. What we now believe is that the secretion of bicarbonate is primarily there to make sure that this accumulation does not happen. And instead, it precipitates those dissolved ions out of solution, and calcium and magnesium carbonate. Once they're out of the solution, they don't exert no osmotic pressure at all. That reduces the osmotic pressure here, which in itself adds a third important mechanism of absorbing water. And that's the reason I actually think that it's fundamentally evolved to do that. At the same time, it does help with removing calcium that it doesn't want to absorb because that can cause kidney stones, so there might be another reason too. In terms of physiology, so I just mentioned the role in water absorption and calcium balance or calcium homeostasis. There are lots of other interesting things we've looked at in terms of roles it plays. One of them is acid-base balance, because you will have noted that for every bicarbonate ion moving from the cell into the gut, there has to be a proton, or acid, moving from the cell into the blood to keep the cell in balance. That means somewhere else has to get rid of that extra proton, and we think now that that's the gills of these marine fish is excreting acid at an equivalently high rate to actually help balance the system. The fourth one is quite intriguing, respiratory gas transport. That's because this bicarbonate all comes from the metabolic CO2 the fish has produced itself. So for every bicarbonate ion and calcium carbonate molecule exiting the gut, that means there's one less CO2 molecule to be excreted across the gills, which is the respiratory organ of these animals. So that means you actually get an anatomical separation of oxygen uptake at the gills and CO2 excretion, most of which still doesn't occur at the gills, but we now recognise that about 10% of the respiratory CO2 of marine fish is actually excreted not in the form of gaseous CO2, but as bicarbonate ions and solid calcium carbonate in the gut, which is quite a novel discovery in itself. So that's as much as I'm going to say about the physiology, unless I can get questions towards the end. I'm going to now turn my attention towards the question, well, how much do these guys produce, and is that important globally in terms of ocean chemistry? Well, the answer obviously is rather a lot. Otherwise, I wouldn't be here. And I can give you some numbers on how much that is, but it's often better to give you some visual indications. Here's one of them. One of the reasons I dressed as a flounder at that party a long time ago was because they are a fantastic animal. Truly wonderful. Most people think flatfish are a little bit boring because they just sit in the bottom and they don't do much. But for me, that's great, especially when you want to take an X-ray picture of a live animal. The other reason, as well as sitting quietly as you put them under the X-ray, is that all these bones show up very nicely on X-rays. That's why we take X-rays of people in hospitals, for example. But in this case, this fish has pretty much no bones overlying the visceral area where the guts are and the intestines. Apart from the pectoral fin here, you can see a little fin rays there, and a few spinal bones there. All the other bones show up, and then you get this nice clear zone. So this, and the third reason flounder are good, is because they're euryhalar. That means they can live in either freshwater or seawater. Now it turns out that freshwater fish don't drink at all, because they don't need to, and they don't produce calcium carbonate in the gut. So this is a freshwater flounder with nothing in the gut at all. What we then did was transferred this freshwater flounder into seawater, took another picture 3 hours later. And what you can see is large X-ray opaque clumps of calcium carbonate crystals forming within the intestines, 2 lumps here, 3 hours after they started drinking in seawater. Let's put that into perspective. This is a 2-year-old animal. So this bone here is 2 years old. It took 2 years to grow. This calcium carbonate took 3 hours to grow. And it's more, well, the same if not more X-ray opaque than calcium phosphate bone. And that's being excreted continuously throughout the life of the animal. Here's another visual indicator. This is our current champion of calcium carbonate production. He's a great barracuda. about 13 kilos. That's quite a story getting it into the tank in the first place. But as you can see, the floor of this tank is completely littered with these white pellets of calcium carbonate. And we siphoned these out, which was, someone had to draw the short straw to do that. And that's how much we collected over a 24-hour period. This is a 50 ml or 50 gram tube. That's about 20 grams of mucus plus inorganic carbonate. Roughly speaking, about half the weight is mucus, so the other half is about 10 grams of calcium carbonate that a single fish produced in one day. When you scale that up, these animals are producing about 2 times their own body dry mass of calcium carbonate per year. That's a lot of calcium carbonate. If you're going to scale that up globally, you need to know all the different factors that are going to affect the production rate within fish. And the most obvious ones that we targeted first of all were temperature, because bony fish are ectotherms, which means they're at the mercy of their ambient temperature, and that controls their metabolism. And the other one is body size, because usually metabolic rate is higher in smaller animals, so we needed to investigate that. And lo and behold, we found the relationships we predicted. And if you look at metabolism alone, the influence of temperature on the x-axis against metabolic rate is usually exponential. And when you do this for fish generally, you get the Q10 value of about 1.86. That means that for every 10 degrees increase in temperature, metabolic rate increases about 1.86-fold. So what we did was we used this relationship for metabolism to help predict what the calcium carbonate production rate would be by fish at different temperatures. We then used the same relationship for body size. Now, ignore that line, because obviously bigger animals have high metabolic rates than smaller animals. But when you scale it to body mass, so per gram of tissue, smaller animals have higher metabolic rates than bigger animals. It's the same reason that human babies have a higher heart rate than adults, they have a higher metabolic rate. That's why my mouse has a much higher heart rate than an elephant. It's just smaller animals, higher metabolism. Same is true with fish. And if you look at this relationship for metabolism within fish, you get about a 1.6-fold increase in metabolic rate for every 10-fold decrease in mass. So we use that relationship too to scale this for carbonate production by fish globally. And you've got a relationship like this that shows body mass on a log scale here, it's carbonate production on the y-axis. And then each of these lines is at a different temperature, going in 5-degree intervals from 0 to 30. And the clear message from this is that the smaller fish at the warmer temperatures is where most of the calcium carbonate is being produced. Now this is all scaled up based on our measurements of carbonate production within the lab of various species. What we now needed to do was take this to the global scale and say how much fish producing globally. And so I began asking the question to ecologists and population modelers that I met all the time. About 2004 I started asking the question, how many fish are there in the ocean? To which no one could ever give me an answer. And in fact they'd say, well, nobody knows the answer, we can't calculate it. Now actually the number of fish is not quite as important as the biomass of fish and the distribution of that biomass across the different size classes and the different temperatures. And then, by chance, I met this guy, Professor Simon Jennings, who I now learned is a collaborator with Trevor, who's a population modelling ecologist, who turned around and said, actually, I think we can make an estimate of this now. We couldn't previously, but now we can. I have a method, let's have a go at it. And he encouraged a colleague of his, Dr. Billy Christensen, up the road at the coast here at the BBC Fishery Centre, to do the same. And this is quite important because these two were doing it using very different methods. And so it's quite important to verify this across different methods to be confident in the result. Now I first asked this question, I think, in 2006, and we began working on it then. And it was about a year later that I got an email from Simon on the Tuesday and an email from Billy on the Thursday of the same week with answers that were dramatically close to each other, which made me think, I think we're on to something. So this allowed us, for the first time ever, to model the biomass of fish in the oceans globally. It was based on breaking the ocean up into 36 by 36 kilometer squares, at least across 90% of the oceans where we have the data. You then combine that with the sea surface temperatures in those regions and our model for estimating calcium carbonate production by fish. You can come up with an answer for calcium carbonate production globally. The global marine fish biomass turns out to be somewhere between 0.8 and 2 billion tons of static biomass. But importantly for us, the rate at which those fish are producing calcium carbonate is somewhere between 40 and 110 million tons per year. Now, what do those figures mean? The only thing that really makes any sense is when you compare that to what the previous estimates of calcium carbonate production from the oceans actually is. When we did that, we got a figure of somewhere between 3 and 15%. And to be honest, even 3% would be quite exciting because no one knew that fish produced this carbonate before, at least on this scale. Having said that, every assumption we did in this modelling was as conservative as it could possibly be. And that's for a reason, that if you're going to try and get this published, you don't want to be seen to be over-egging it and exaggerating the situation. So every point where we had an assumption to make, we made the most conservative possible assumption. So this really is a very conservative estimate. And in fact, if you apply realistic but less conservative assumptions, this figure could easily be 3 times higher. So it could be somewhere between 9 and 45%. If you ask me to bet on it now, I'm guessing it's going to come out closer to 45 than 50%. Some of the reasons for that are all the conservative assumptions so far are based on using data from non-fed fish. We know that as soon as you feed them, they produce even more. It's based on inactive fish sitting in tanks doing nothing. And we know that if they're active and swimming, they drink more, they produce more calcium carbonate. It's also based largely on benthic species that are known to be less active than pelagic species. So, given that the metabolic rate is 2 to 4 times higher in fish that are active feeding pelagic, this could easily be 3 times higher on a global scale. And actually, we now know things I will show you later on that make this figure could be even higher again. So there's a very large amount of calcium carbonate coming out of fish in the oceans globally. That's an interesting story. But it's only part of the issue, because what happens to that carbonate once it's been produced is just as important in controlling the ocean chemistry. Now, calcium carbonate sinks because it's so dense. If it sinks to the bottom of the ocean and sediments out down there, then it's removed that carbon from the site. But calcium carbonate is unlike a lot of salts in that it's more soluble in cold water than in warm water. So as it sinks to the cold ocean depths, it's more likely to dissolve. The pressure as well is also acting to increase the solubility. So this reaction is more likely to be reversed and the calcium carbonate to dissolve as it sinks within the ocean. When that happens, it increases the alkalinity of the oceans from the surface. And it turns out that this is only supposed to happen for calcite and aragonite once you get down to fairly deep depths, down to the saturation depths. Fish carbonates, on the other hand, I've already mentioned, has much higher magnesium content than most other calcium carbonate products, somewhere between 10 and 33% of the best estimates we've had at the time. And in theory, this should make this type of carbonate much more soluble than either calcite or aragonite. So could it be that this fish carbonate is actually dissolving at much shallower depths than other types of carbonate? Well, this would be very interesting if it's true, because it might help explain a phenomenon that's been known for several decades within ocean chemistry that's not been easy to explain. And that is, rather than the seawater alkalinity increasing at its fastest rate once you reach the saturation depths for either aragonite or calcite, it actually seems to increase at the fastest rate over the first 500 to 1,000 meters, which can't be explained by the normal solubilities of aragonite or calcite. So could it be that this effect can be explained by the high solubility of the magnesium content of the fish carbonates, causing this large amount of calcium carbonate to actually dissolve within the first kilometre of ocean depth, increasing the alkalinity over the range that is seen. So the fate of this calcium carbonate is very important to know. At the moment, that is speculation. based on the magnesium content only. We're yet to do any work to actually work out whether this is actually happening or not. Now the media got hold of this publication, and they really like short, snappy titles of fish poo saving the world, which, let's face it, is a bit of an exaggeration. It's also not true. And it's not true for at least two reasons. The first one is, it's not poop. Because it's a subtle comment, but it's not undigested food. It's not fecal matter. This is endogenously produced carbonate from the fish's own metabolism. So that's a subtle bit of wrong. The other reason it's wrong is they're not really saving the ocean. And that's because, what I said at the start, any calcifying organism is actually contributing to ocean acidification. Because the generation of calcium carbonate in the first place produces CO2. Having said that, fish poop, one of the better words, might be less of an evil than other forms of calcium carbonate. Because if it dissolves in shallow waters, at least this process is reversed, and the net effect is neutral within the surface oceans. So you could say that fish carbonates may be less damaging than traditional carbonates with respect to the marine and atmospheric CO2 situation. So it doesn't matter how often you tell the media you got it wrong, they still like those connections. The point to make here is all the work I've mentioned so far was done with no funding whatsoever. That's not quite true. The fundamental physiology was, but all of the global carbonate stuff, no funding whatsoever. Because the NRC, who've now funded this, starting last summer, I don't think they believed it. But once you get it published, and the figures are big and interesting, and oh, maybe it's worth funding. So now they've funded it, we're going to now go back and do more precise estimates based on the less conservative assumptions and get some hard figures on that. And so in a couple of years' time, I should be able to tell you the full answers on that. What we're going to be doing as well is actually looking in sediment traps from around the world to see if we can find evidence for fish carbonates at different depths. We're going to actually measure the solubility of the fish carbonates produced at different temperatures by different species, so we can again put hard numbers on the actual solubility issue, which is obviously very, very important. One part of this picture I haven't said anything about yet is the opportunity for fish carbonates to sediment in shallow regions. And in fact, this is a very interesting question because the tropical shallows are somewhere where it might actually be preserved rather than dissolving. And that's because, being in the tropics, it's warm, and that reduces the solubility of calcium carbonate, as I mentioned. Being the tropics and shallow, it means it's low hydrostatic pressures, which again reduces the chance of it dissolving. And thirdly, there's usually lots of fish with high metabolic rates in these warm tropical areas. So I wanted to look at this question of whether fish carbonates could become part of carbonated sediments in such areas for a long time. But I had to search to find people who are interested in joining me, particularly geologists. And this is where an element of fortune happened here, where I met Professor Chris Perry from Manchester University. And I have to give credit here to a little-known airline company that runs out of Exeter, where I'm from, because I was flying back from a meeting in Manchester on a 70-seater plane on flightly.com. There were 5 people on this 70-seater plane. And I thought, great, I'll get a seat to myself, and I can sleep or get on with work or whatever. And to my horror, I found that they'd sat me next to one of the 5 and I thought, well, I can sit here and be grumpy and get on with it or not. And then I noticed he was reading a scientific manuscript, and I kept seeing the word coral reef, carbonate, sediments, tropics, and I couldn't resist. I said, excuse me, could you tell me what you're reading? And then he told me, I'm a marine carbonate sedimentary geologist. And so I happened to have my laptop open. I showed him some of my pictures, and by the end of this 40-minute journey, he was absolutely gobsmacked. And he said, you know what, this could transform my field. We need to work on this. So that was a Thursday evening. I drove him to his hotel where he was staying. We met the following lunchtime and agreed to write a grant proposal that we submitted the following Monday. Now, it's not quite as tidy as that because we didn't get that grant. But we were sufficiently encouraged by the feedback that we wrote another one to NERC and they funded it. And we used that to go to the Bahamas to answer these questions. So we went to a great place called Cape Luther Island. It's great when you can get Research Council funding to do this kind of thing. Which is in the Bahamas, warm tropics, the most likely preservation site for this kind of carbon. And there we collected local fish species of a variety of types in different habitats. We brought them into the marine labs at the Cape Luther Island Institute and collected the carbonates they produced, measured them, and then had a look at their morphology as well. We also went out into the local habitats and collected the organisms that were traditionally thought to provide most of the carbonate sediment in these areas, which is mainly calcareous green algae, such as Halimeda and Bacillus. When you collect these organisms and digest all the organic material, you're left with inorganic calcium carbonate. So these animals have 2 to 4 crops per year. Sorry, these algae have 2 to 4 crops per year. These have about 5 to 6 crops per year. And that's what an individual will produce during one particular crop. What this shows you is the amounts fish produce on the right compared to these algae on the left. And again, these are small numbers of individual fish, but these are producing them every 24 hours. These are producing them a few times a year. So visually, it gives you a scale of the amount of carbonate being produced by fish in these areas, which has the potential to be a lot. So we then coupled together with some ecologists who were able to do we did the calcium carbonate excretion rate measurements, and they did the fish species abundance surveys. The other thing we noticed while we was out there, remember I talked about metabolism scaling with body size? Well, when we measured that relationship between body size and calcium carbonate excretion rate in these tropical fish, we found an even higher scaling relationship with body size, such that with every 10-fold decrease in body size, you get almost exactly a doubling of calcium carbonate excretion, which compares with only a 1.6-fold increase for every 10-fold decrease in body mass from metabolism. So calcium carbonate production actually scales even more greatly with body size than for metabolism alone. We then went out into the field to measure or look at data that was already available on fish species abundance across the 7 different habitat sites within the Bahamas, and found some really quite staggering numbers when we put it back together with our calcium carbonate measurements in the lab. In some places like mangroves, fish were contributing the majority of calcium carbonate. In others, like Gorgonian hard ground, up to a quarter. In some places, obviously, there's lots of other producers producing the most of it. But on average, across the whole of these 7 habitats, fish are producing about 14% of the total calcium carbonate, which is, again, a surprisingly large figure. Back to the question of what these looks like, and I show you this picture of the rugby ball or American football shaped crystals. When we looked at a lot more species, we found it was actually quite diverse. We found things that looked like those ellipsoids, things that looked like straw bundles, dumbbells, setting spheres. I called it broccoli. Equivalent of broccoli, large spheres. Different species produce quite different looking crystals. It's quite possible that there's a relationship, a temporal scale relationship, where the bigger ones are produced the longer the intestine is, for example, or bigger fishes. We're yet to investigate that fully. The bottom line is, although species produce quite different looking crystals, they're all fairly unique. They're not completely unique. They're all fairly unique relative to other carbonate crystals produced in the marine environment. So we then thought, is there any chance of going out there and finding them, indeed finding the carbonate needle in the marine sedimentary haystack? So that's what we did. We went to all of these seven habitat types, dived or snorkeled, collected sediments from the bottom and had a look. And lo and behold, the answer is yes. We could find the needle in the haystack. In fact, in all 7 of these habitat types, we found abundant evidence of fish carbonate-looking crystals within those sediments, which was just published last week in PNS. So that's quite an exciting result, which resulted in a lot more media attention about fish poo instead of Finally, I'm going to talk about ocean acidification. You will have heard lots of talks about the acidifying ocean being more corrosive to calcium carbonate, and that's undeniable. But actually the net effect on biogenic calcium carbonate production will depend on the relative changes to both biogenesis in the first place and its erosion. Now, it turns out that whilst the prediction was that calcium carbonate production would go down in an acidified ocean, it actually varies a lot. Some animals, some organisms decrease, some increase, some don't change, others are biphasic. And there's more and more data now coming out showing this variety of responses. What we can say is that for fish, we have some very clear predictions about what's going to happen to carbonate production. And we just had an ocean acidification grant funded to look at all sorts of effects of CO2 on fish, including their development and growth, but also, I'm happy to say, to look at their carbonate production and how that might be effective regarding the carbon cycle. Our prediction was very simple, which is that carbonate production by fish guts would increase in response to ocean acidification. There's two reasons for that, and those are that if you expose fish to a high level of CO2, the level of CO2 in the blood rises. That in itself has a knock-on effect of increasing the amount of CO2 in the cells of the gut, and that will fuel more bicarbonate production in the intestine. Secondly, the fish's response to ocean acidification is to increase the buffering in its blood by actively accumulating more bicarbonate ions. That in turn fuels more bicarbonate directly through the same transport system. So there's two reasons why we would expect bicarbonate secretion to increase in the intestine, and that to end up increasing the amount of calcium carbonate we produce. We've done one experiment so far to measure that in my favourite species, the European flounder, and that has confirmed that if you expose fish to a high level of CO2, you increase the CO2 in the blood, the pH gets well defended, and that's because they increase the amount of bicarbonate in the blood as well. Don't worry, I am aware this is well beyond an end of the century levels of CO2. This was a physiological experiment just to understand the process. When we did this and then measured the gut fluid chemistry, we found, first of all, the gut fluid bicarbonate increased quite substantially in these same fish, and lo and behold, the amount of calcium carbonate they excreted more or less doubled within this range. Now then, 4,500 ppm is well beyond end of the century predictions. But if we assume there's a linear relationship between the amount of CO2 in the water and the amount of calcium carbonate excreted, we can make the following predictions. And those are that if it increased to about 750 ppm, we'd see a 13% increase. At 1,000 ppm, a 21% increase. 1,500 ppm, 39% increase. I'm quite pleased because this matches the estimates based on some in vitro work on a completely different species by colleagues in Miami. This figure is exactly what they got by measuring it in vitro as well. So I'm feeling comfortable that we're close to a realistic prediction about the effect of CO2 on its own on calcium carbonate production by fish. Now, CO2 might be going up, but it's not going up in isolation. Temperature is increasing as well. So understanding the relationship between temperature and carbonate production is equally important. Now, I mentioned earlier that the metabolic rate increases about 1.86-fold for every 10 degrees temperature increase. We've now measured carbonate production in different fish species over temperature ranges, and we get much higher Q10 values. It increases by somewhere between 2.3 and 2.8-fold for every 10 degrees increase in temperature, which is way more than metabolism. If you put that together with the effect of CO2, then if we look at an end-of-century scenario of 4 degrees plus and 750 ppm CO2, we predict carbonate production by fish to increase by more than 70%. Most of that being due to temperature rather than CO2, but it's still 70%. The other observation we made is that the magnesium content increases quite steeply at high temperatures. And the more magnesium there is, the more soluble this calcium carbonate tends to be. So you have a tendency for much more reduction and much more soluble. This is stuff that we're about to submit to proceedings with all society. So, to conclude, physiologically we think calcium carbonate excretion production in the gut is important for water balance, calcium balance, acid-base balance, gas exchange. We think it's important for ocean chemistry as well, with respect to the fact that we think there's a lot being produced by fish that's globally significant. It's likely to dissolve more rapidly in the open oceans, but we need to confirm that now. There's also potential for it to be preserved in sediments, and that's something we want to look into, at least in shallow tropical environments. And regarding future predictions, it would be very simply that global warming would increase the magnesium content and make it more soluble, as well as increasing the amount. And CO2 would do the same, and the two are likely to act synergistically. Final slide. Fishing impacts. I'm not going to say much about this at all. Simon Jennings has done some modelling on this. Essentially, as you remove larger fish, you tend to be leaving smaller fish behind. They're producing most of it anyway. So calcium carbonate production, we predict, would fall less than expected based on just population biomass decline. But importantly, the modelling also suggests that calcium carbonate production by fish falls rapidly at relatively low rates of fishing that are used as management targets currently. So if you're concerned about the carbonate production by fish, it's an issue. So the bottom line is fish physiology has some influence on the control of ocean chemistry, or potential to, and it may be one more additional factor to consider when assessing climate change and fishing impacts on fish populations. I'll finish by thanking lots of people, as there always are, involved in such research. All of these people are colleagues and students of mine at Exeter, and then the whole list of people who are involved in this research in the past. And funders, BBSC have funded all my fundamental physiology work, NERC, and more recently being convinced to fund the environmental activities as well. Both sides are welcome trust to fund small pockets of work too. And I'll finish by saying thank you, and any questions. And I'll leave that as a provocative slide. As the calcium carbonate contained rock, if you want to ask me about where that might come from.
There's time for a few questions. I'll leave Rod to pick and choose the people who look like the easiest question on the basket.
Can you tell us, of the total amount of fish production, how much of that fish production, what percent of that fish production in the open ocean as compared to the coastal ocean? Is most of the calcium carbonate production that you're talking about actually in the coastal ocean?
Yes, most of the calcium carbonate production by fish is where most of the fish are, which tends to be around the continental shelf. So yes, that's where most of it is. If you looked at the global map I showed of fish population biomass, it is concentrated around the continents, yes.
Okay, so an alternative hypothesis for the alkalinity balance that you tried to show us could be that instead of dissolution in the open water column of the fish poop, or the calcium carbonate, isn't it possible that this is actually an advective process of the disaggregate fine-grained calcium carbonate materials off the shelf in the open ocean in dissolution? You have to get some way where fine-grained particles are dissolving. And it would seem to me that you have more material that's disaggregated and then being advective out to change, dissolve in a way that has enough time.
That's a very good point. So is it sort of sideways movement of continental shelf deposited material moving out into the open ocean? Is that what you're... Yeah. And if it really is fine-grained stuff, then it's going to stay suspended for longer and has more chance of being delivered horizontally. Yeah, I like that idea. Yeah, we should talk about that. Yes.
If fish are producing all of this carbonate, do you see that it corresponds in the fossil record to fish proliferation?
Excellent question. So this has been something that has tantalized me for a year or so now. And I got a couple of an undergraduate student, Jacob Bill and Rebecca Costa, and the current postdoc Aaron Regan involved in trying to answer this question. So could ancient fish produce sufficient carbonate to be part of the fossil or carbonate rock record? Well, one thing I haven't mentioned yet is that ocean chemistry has changed quite a lot in the geological past, in particular in relation to calcium concentration, or calcium and magnesium and sulfate. And in fact, calcium has gone up and down, and sulfate and magnesium have gone up and down reciprocally. So that if you look about 100 plus million years ago in the Cretaceous period, calcium was 4 times higher than it was today. Now, we know from experiments we've done with fish that if you add more calcium to the seawater, they produce more calcium carbonate. There's more being drunk, whatever. The other thing is, when did teleost fish evolve? It was kind of around, they were really radiating around the Cretaceous period. So we know there were lots of teleost fish around, we know the calcium levels were higher. So this induced us to do a time machine experiment. We took modern day fish back to the Cretaceous period, we didn't really. What we did is we made some seawater that was artificial, that matched the Cretaceous seawater chemistry as far as we could get. And when we put 3 different species under these conditions, we got anywhere between not just 4, but 4 to 10 times as much calcium carbonate. Also, when we did that, the magnesium content reduced dramatically 2 to 4-fold, which would again increase its preservation potential within sediments and ultimately rocks, and much more to produce. When we looked at the crystal morphology, it's completely different. It changes from a spheroid type structure to this really blocky cuboid type structure. So I don't know the answer to your question, but I'm really intrigued to take this a little bit further. Having done sort of lab type experiments to go out into the field, look at samples from the past, and see if we can get a signature from the chemistry of these carbonates and see whether they match what we're seeing within those sediments. Having said that, I've talked with some paleogeology people, and when they're chipping away at the white cliffs of Dover and things like that, to collect the coccoliths to look at their chemistry, I said, what's around it and what is it? And they said, well, about 50% or more of what we collect, we can't identify. And I said, well, what size crystal is it? And they said, it's about 1 to 2 microns, which is the same as our crystal. So anecdotally, we potentially are onto something quite interesting there. That it could be that the cement surrounding the occolith fossils is actually fish poop. You've got me saying it.
I'm thinking of bioenergetic equations, and I've never seen them contain a component that represents the mucus bubbling calcium carbonate. Is it a trivial thing to do that energetically, or does it take a lot? You said it's half the mass or something.
Yeah, it's about, yeah, but half the mass is the mucus that also contains a lot of water.
Okay,
So the amount of dry matter is probably pretty small because there's a lot of water in that. So I would guess that energetically it's pretty small, and that most animals, whether they're fish producing this carbonate or not, your intestinal lining is producing mucus all the time as a protective measure. So it's probably a normal part of all animals' physiology. It's just we see it very clearly in a non-fed fish that's producing these very visible white crystal-containing mucus. But I would guess it's pretty small. We could probably estimate that if we've got numbers on weights if you're interested.
So the related question: how long does that mucus stay attached to those crystals, and does that impair its ability to dissolve?
Okay, so how long does the mucus last? Well, that's an important question to ask regarding if this stuff is sinking or if it's staying in suspension for long, how long before those crystals are released? If you just keep them in a beaker and aerate the beaker to mix it, within hours it's breaking up into small particles. Within days the mucus has kind of been metabolized by microbes. But we're doing that more thoroughly now, so we'll get a proper answer on that soon, but it's hours or days.
You've said a couple of times that these are non-fed fish. How do you expect eating fish to differ?
So we have measured carbonate production in feeding fish, and they produce the same amount or more. The other thing to take into account is what they're eating. So diets vary enormously in how much calcium they've got in. If a fish eats a ragworm, which is a soft-bodied invertebrate, there's very little calcium in it. If it eats a crustacean that's got a calcium carbonate shell, there's tons of calcium. If it eats a fish with a calcium phosphate skeleton, it's got tons of calcium. And what happens is it goes into the stomach, which is acidic, which completely dissolves all the bone or the shell of the animal, releasing all the free calcium, and then that enters the intestine, which then gets precipitated. So we don't have the numbers on it yet, because that NERC grant is beginning to answer that question about how the diet itself affects the carbonate production rate and the types of carbonate that produce. But we reckon we'll see potentially a massive difference depending on the diet they're actually eating. But we suspect it'll be higher in feeding animals most of the time.
Time for one more question? Everybody's ready for the light reception.